Here are natural polymers, which is lesson 22.6. We will be going over the types of proteins, the building blocks of proteins, amino acid structure and terminology, carbohydrates, and the genetic process. So let's get started. Proteins are natural polymers that perform many functions in the human body. There are different types, like fibrous proteins and global proteins. Fibrous proteins are proteins that have structural integrity and strength for many types of tissues, like muscle, hair, and cartilage. Global proteins are rough, spherical-shaped proteins who transport and store oxygen and nutrients, catalysts for vital functions, regulate body systems, and transport electrons to metabolize nutrients. The building blocks or monomers of protein are called alpha amino acids, which are from the amino group NH2, which is being attached by the alpha carbon group CO2H in the example. R shows H, CH3, or a more complex substituent. The R groups are side chains that determine if an amino acid is polar or nonpolar. Polar side chains are hydrophilic, which contains large numbers of nitrogen and oxygen atoms. Nonpolar side chains contain mostly carbon and hydrogen atoms and are hydrophobic. To the right are a list of common proteins, which contain nonpolar and polar R groups. Peptide linkage is a chemical bond formed during a condensation reaction between a molecule with an amino group reacting with a molecule with a carbonyl group. Dipeptide is a product of a condensation reaction that contains a peptide bond. Like in the example, you can see that the product is in the middle in a box, which is a peptide linkage. Polypeptide is a product of additional condensation reactions to yield a protein.
Consider this a tripeptide because it's in between three groups, opposed to a dipeptide, which is only in between two groups. Next, what we're going to be talking about is the secondary structure. And the secondary structure is the arrangement of the chain of the protein molecule. And then the secondary structure is determined by the hydrogen bonding between the lone pairs of the carbonyl group's oxygen atom and the amino acids group's hydrogen atom, which is bonded to the nitrogen atom. And these interactions form the alpha helix structure, which helps with elasticity. And then the alpha helix, when it breaks down to a secondary configuration, it's known as the random coil arrangement. And then the interactions within different protein chains, they form a pleated sheet arrangement. And they indicate a strong structure, but they are resistant to stretching. The alpha helix structure, they have intra-chain bonds, while the pleated sheet arrangement, they have interchain bonds. Next, we have the tertiary structure, and it is just the overall shape of the protein. And then the tertiary structure, it is maintained by the hydrogen bonding, the dipole-dipole interactions, the ionic bonds, the covalent bonds, and the London dispersion forces between the nonpolar groups. And to stabilize a tertiary structure, you use the amino acid cysteine, and it's used to oxidize and form a disulfide linkage, which is an SS bond. And then we have something called denaturation, and it is the process of breaking down the 3D structure of a protein. And the denaturation affects the function of a protein by breaking down its secondary and tertiary structures. And by breaking those structures, they break down the arrangement and overall shape of a protein, and that can uncoil the helix and disrupt the sheet.
So now we are going over carbohydrates. Carbohydrates serve as a food source for most organisms, and they are structural materials for plants. Their monomers are monosaccharides, which are simple sugars, and they are polyhydroxy ketones and aldehydes. If we were to use glyceraldehyde as an example, it shows that it has a tetrahedral arrangement, and that structure will show that there are 4 different groups that are attached to the carbon atom. Because of this, it shows that there it would always have a non-superimposable mirror image. And that would give a rise to a pair of optical isomers. And in this case, glyceraldehyde would have 2. So this chart right here shows the most important pentoses and hexoses. So pentoses contain 5 carbon atoms, and hexoses have 6. All of these monosaccharides are optically active, and they generally have one isomer that is more common in nature than the others. If these monomers were to be in aqueous solution, they would be in a cyclic structure, which is basically a ring structure. If we were to combine these monosaccharides, they would create a more complex carbohydrate. For example, sucrose. Sucrose is a disaccharide, which is sugar that has 2 monosaccharides. These are formed from glucose and fructose, which is through a process called glycoside linkage. In this process, it eliminates water in order to form carbon to oxygen to carbon bonds between the rings. Now we are moving towards polysaccharides, which are large polymers that contain a lot of monosaccharide units. 3 main important polysaccharides are starch, cellulose, and glycogen. Starch is a polymer of alpha-D-glucose, which contains 2 parts, amylose and amylopectin. Amylose is a straight-chain polymer of alpha-glucose. However, amylopectin is heavier, and it is the highly branched polymer of alpha-glucose. Branching occurs when a third glycoside linkage attaches to a branch to the main polymer chain. Moving forward, cellulose is a polymer of beta-D-glucose, and those linkages in cellulose give the glucose rings a different relative orientation compared to the starch. So even though it is a minor difference, it is still a very important change. Similar to the amylopectin structure is glycogen, but it has more branching. And these branchings allow the glycogen to break down into glucose when energy is needed. So here is a picture showing the difference between starch and cellulose. So if you can see, the positions of the oxygens are different, and the way it is structured is different too. But even though they may seem like minor changes, like I said before, they are still very important to know. Now we're going to shift towards nucleic acids, where we're going to learn about DNA and RNA and their processes and differences. DNA is a polymer that stores and transmits genetic info, and it is placed in the nucleus. And RNA is found in the cytoplasm outside the nucleus, but it is smaller than DNA. Nucleotides are monomers of nucleic acids, and they contain 3 parts. They have a 5-carbon sugar, a nitrogen-containing organic base, and phosphoric acid molecule. If you want to use a reference, you can use the images to the right, which are the first 2 parts. In order to function, DNA has a double helical structure with complementary bases on the 2 strands, and these bases form hydrogen bonds to each other. Cytosine and guanine will always be a compatible pair for the DNA strands, and same goes with thymine and adenine. So the replication of DNA is one process that allows for the transmission of genetic info as the cells divide. And another major function is protein synthesis, which is a major function. A gene is a given segment of the DNA, and they contain code for a specific protein. And these codes transmit primary structure of protein to the construction machinery of the cell. And each specific code for each amino acid ensures that the correct amino acid will be inserted as the protein chain grows. And a codon is a code with a set of 3 bases. As DNA contains the genetic information, RNA transmits that information to the ribosomes, where the actual protein synthesis process occurs. And messenger RNA is built in the cell nucleus on the appropriate section of the DNA. And it looks like an unzipped double helix. Complementarity of the bases is used in a similar process like the DNA replication. And after that, the RNA migrates to the cytoplasm. and there protein is synthesized with the assistance of ribosomes. Transfer RNA is basically small RNA fragments, and they're to find specific amino acids, and then they are told by the codons to attach those amino acids to the growing protein chain. And then they are to break the code of the genetic message from the mRNA using an anticodon. Protein is built in a series of steps. So first, the tRNA molecule brings an amino acid to the mRNA. The second step is that another tRNA moves to the second codon site of the mRNA with its specific amino acid. Then two amino acids will join together through a peptide bond. And after that, the tRNA on the first codon, breaks away. And after that, the process just repeats. And the tRNA anticodon will always match with the mRNA codon.